Very welcome to the Max Planck Institute for Biology of Aging in the city of Cologne in Germany. My name is Gabriella Lundkvist, and I am now standing here in our beautiful institute building, which was inaugurated in 2014. This evening we are delighted to have you tune into our special age art program with our special guest tonight, Professor Ueli Schibler from Geneva, and the American pianist Hilda Huang. The topic of tonight is rhythms. We will hear beautiful musical rhythms written by Bach and Beethoven, but also we will hear about the rhythms in our bodily functions, the so-called circadian system. Have you ever experienced jet lag? Or did you notice a change in your sleep when we switched to so-called winter time recently? Well, if so, I can tell you why. This is because our bodies have an inner clock that tells us what time of the day it is. And when we move rapidly across different time zones, or when we shift the time 1 hour in spring and fall, the inner clock is no longer in sync with the outer environment. For instance, you have just landed in the afternoon in New York, but your body is still on Germany time, and you just want to go to sleep. So this causes chaos in the organ functions, and then we experience what we recognize as so-called jet lag symptoms, which can be very frustrating. So what does the body clock do? Well, it regulates all our bodily functions to make sure that the right function is turned on or turned off at the right time of the day. For instance, the blood pressure and the heart rate increases during the day. Certain stress hormones are produced during the day when we are awake, whereas our immune system becomes very active during the night, and other restoring hormones are also released during the night when we sleep. The master clock in our body is located in our brains, in an area called hypothalamus. And this clock knows what time of the day it is because it receives light through the eyes. But this is not the only clock that we have. Ueli Schibler and also other scientists, they discovered that we have clocks everywhere in our bodies. We have it in the skin, we have clocks in our liver, in our heart, in muscle tissue, the ears, the bones, basically everywhere. So how do all these clocks work together? How do they communicate with each other? Well, Dr. Schibler, he explains it to us as this is like a big symphony orchestra with many different instruments. And each instrument can play by itself. But the orchestra needs a conductor that coordinates and leads the individual instruments. I am pleased to introduce to you tonight the Circadian Symphony by Professor Schibler.
Ladies and gentlemen, I'm extremely pleased to talk to you about my passion, which is our endogenous clock system. I would like to thank the organizers, Adam and Helen, for putting this very nice event together. I'm particularly honored that I can do this and participate in this event together with such a talented pianist, Hilda. Well, the two subjects, music and the circadian timing system, are at least related in two aspects. Both deal with rhythms, and in the case of a symphony orchestra, as shown here, both are organized in a sort of hierarchical fashion. In a symphony orchestra, we have a director, and we have musicians that listen to the pace of the rhythm that he gives, whereas in the circadian timing systems, that, as you will see, regulates most of our behavior and physiology, the director is a clock, a large clock, which gives the pace to the musicians, and the musicians in this case are clocks that exist in virtually all body cells. What is the circadian timing system about? We all know about the geophysical time, and the day, as we all know, is defined as one rotation of the Earth around its own axis. What is circadian time? Well, circadian comes from circadium, which Latin names, which mean about the day. And depending on the species, our timing system can measure time only approximately between 23 and 25 hours. But how do we know that there actually is a clock, or there are clocks? There's a very simple experiment that can show this. You just have to place a mouse, in this case, or a hamster or a rat, into a cage equipped with a running wheel. And these small rodents, they love to run the wheel. So the moment they wake up, they enter the wheel and they run a lot. And this mouse here that you see ran about 15 kilometers every night. Now keep in mind that mice are nocturnal, not like we are diurnal. So mice are active during the night. Now if you measure the revolutions of the wheel, of this running wheel here, there is a software that converts these into so-called actograms. And this is shown here. So what is the actograms? It just shows you the activity bouts in black. And you see then when the animal is exposed to light-dark cycles, it runs during the dark phase, where it's very black here. In these actograms, for graphical reasons, one actually shows day 2 after day 1, and then we re-put day 2 here, and this would be day 3, day 3, day 4, and so on. This is just for graphical reasons. So we see 2 days. It's clear that the mouse runs during the night exactly in a 24-hour rhythm because it's exposed to light-dark cycles. Well, that doesn't mean that the mouse has a clock, because it could just react to the light or to the dark, meaning that it prefers the dark and runs during the dark. So how do we know there is a clock? This is shown below here. Because if we shut off the light and keep the temperature constant, so there are no signs of time from the outside world, the mouse is just as rhythmic. But as I mentioned 2 minutes ago, each day of the circadian time is, in this case, a bit shorter than a real day, about 23.7 hours. For humans, it would be 24.3. So this actually advances because each day is a bit shorter. Nevertheless, the mouse would keep this very strong rhythmicity for its entire lifetime. It's a very robust system. Well, is this system important? It is, because it regulates the timing of most of our physiology. As we just have seen, rest-activity cycles, but it also affects the rhythm of the cardiovascular system. So even if you stay in bed all day long, your heartbeat frequency and your blood pressure would oscillate in a daily fashion. Body temperature has a rhythm, very narrow rhythm. It oscillates between about 36 and 37 degrees. So you can say, well, what's the importance of that? It is important because it actually helps to synchronize the body clocks in all our peripheral tissues, like the liver, skin, and so on. Well known, the endocrine system. Most hormones are secreted in a daily fashion, some more than others. The most extreme case is melatonin, produced in the pineal gland of the brain. If you measure it in the blood, it oscillates 100-fold every day in the blood. But glucocorticoids, which are made by the adrenal gland, in the case of humans, cortisol, oscillates also about 10-fold. Then visual accuracy oscillates during the day. There are times during the day where you see better than other times. Kidney, renal system. It makes sense that the kidneys produce much more urine during the day than during the night. This lets you sleep throughout the night. You, I say, and I mean younger people than me, because for older people, as you will see at the end, the system degenerates and the rhythms are less robust. So older people occasionally have to stand up in the middle of the night to go to the toilet. Activity of the digestive tract, for example, hydrochloric acid is secreted into the stomach in a circadian fashion. This makes sense because you don't want to have hydrochloric acid in your stomach if there is no food there. This would actually attack your cells. Then the peristaltism of the gut to transport the processed food is also highly circadian. And the liver is almost the extreme case. The liver is responsible for a lot of our metabolism. It is responsible for fat metabolism, for protein metabolism, for sugar metabolism. Very importantly, it's also important for detoxification. Whenever one eats, one also ingests dangerous stuff, toxins. And these toxins have to be eliminated by the body. So the liver is equipped with a system, an enzyme system, that makes these toxins more water soluble so they can go to the blood and be secreted through the kidney. I said it's probably the most extreme case of circadian organs because we have recently discovered that in mice, the liver, the entire liver, its weight and the cell size of the liver, so the hepatocytes, and the macromolecular content, so the content in proteins and lipids, oscillates by 30 to 40% every day, meaning that the liver actually gets bigger and smaller during the day. And there are indications that this also applies to humans, although the amplitude may be a bit lower than 30 or 40%, perhaps 20%. So this system is very important because it defines the timing of virtually all our body functions. I said in the beginning, it is organized a little bit like a symphony orchestra. It has a master clock called the master pacemaker in the suprachiasmatic nucleus. It's actually two, it's nuclei. These are two very small structures. Diameter in humans, about 3 millimeters, in mice about 0.2 millimeter, only composed of a few thousand neurons. There is a very precise method to lesion this structure in mice or hamsters or rats. It's called the stereotaxic lesion, where you can actually burn out 1 millimeter of the brain which contains this structure, and then the animals are completely arrhythmic. So the master pacemaker, we know, is at least important for the rest activity cycles. We'll talk about the peripheral clocks a little later. So the master pacemaker is composed of neurons, and it has been shown that each neuron has its own clock. So it's actually a cellular quality. But the clocks are not that precise, so they have to be synchronized among themselves. So they talk to each other via chemical pathways to keep the synchrony within the SCN. So it is believed that the SCN sends out rhythmic signals. I said body temperature, but metabolites, hormones, and in some cases the peripheral nervous system, PNS, in order to synchronize all these countless clocks in the periphery. I will show you that metabolites are probably very important because feeding fasting cycles, of course they also regulate some hormones, but feeding fasting cycles are the most dominant Zeitgebers or timing cues to synchronize the peripheral clocks. The central clock must be synchronized to light-dark cycles so as to stay in harmony with the outside world. Curiously, some blind people, even though they can't perceive light, are nevertheless synchronized, and this probably happens through social cues, being together with other humans that have a normal rhythm. But a lot of blind people actually free run. Free running means they're just following their own clocks, and they have days of 24.3 hours instead of 24 hours. And this is of course a problem because after a certain time they're always in disharmony with other individuals. How do these cellular molecular clocks function? This is a very bit complicated to explain for non-molecular biologists, but I try. I said they exist in all cells. Here you have a cell. The cell, of course, the cell has a nucleus, and the nucleus contains all the genetic material, our DNA. Our DNA is written in a 3-letter code, so 3 letters, in this case 3 nucleotides, determine one symbol or one letter in the writing. So in order to be expressed for a gene, it has to make a protein, and this takes several steps. The most important ones are transcription and translation. So in the first step, there is an enzyme called RNA polymerase that copies one strand of the DNA into RNA. This RNA is then cleaved and modified chemically to produce the mature messenger RNA. The mature messenger RNA is leaving the nucleus, goes into the cytoplasm, where it encounters the ribosome, which is a big macromolecular machine that can translate the 3-letter code, 3 nucleotide codes, into a code of 20 amino acids to make a protein. So this is for all proteins. Now let's assume that you have a clock protein, a clock repressor protein as they're called, shown here. So what happens to generate this oscillation of its accumulation in the cell is that it feeds back in a negative feedback loop on its own expression. That means that once the concentration is high of this protein, it goes to the nucleus, binds to its own gene, and inhibits the expression of the gene, the transcription of its gene. Since there's no longer transcription, production stops, the clock proteins will decay until they're no longer sufficient to inhibit their own expression. And that means that a new cycle can start. This is the way we think these molecular oscillators work. This shows it in a little bit clearer way. So one has these important repressor genes whose proteins accumulate in a circadian fashion. And there are activated genes. You need an activator so the gene is active, but they're less important here. So the activator makes an activated protein. As a consequence, the gene is active, the repressor gene. The protein is symbolized as Rs. So the repressor proteins augment in concentration. When they have reached a certain concentration here, they remove the activator and repress the gene. So now it's no longer transcribed. The curve goes down, and through a mechanism that we don't quite understand, this generates nearly 24-hour rhythms. Not precisely, but close to a few minutes. We also know some of the components, the most important ones, I believe. There is a primary negative feedback loop, and the activated proteins are B, MAL, and clock. You can forget about the names. And there are 4 repressor proteins are period 1, period 2, cry1, cryptochrome 1, and cryptochrome 2. This loop is actually stabilized through a secondary feedback loop that is linked to the first one. So the first feedback loop regulates the oscillations of these repressors ref verb alpha that repress the activators of the activated gene. So this generates a secondary loop that makes the major loop more robust. I was talking about the master clock, the director of the orchestra, which is the suprachiasmatic nucleus. I've already told you that it is composed of a few thousand neurons that are connected. And this system is so robust that one can even visualize it in vitro tissue culture. So you can actually take a slice of the brain which contains this little master clock structure, the suprachiasmatic nucleus. And if you take this slice from the brain from a mouse that expresses firefly luciferase, you know that fireflies produce light during the night, then this luciferase will oscillate during the day because it's regulated by a clock gene, in this case PER1. But of course mice are not, or hamsters or rats or we are not fireflies, so we need a substrate of the luciferase enzyme which is called luciferin. So one has to add the luciferin to the tissue culture medium. And now we can place these below a luminescence microscope and film it through many days. And what you will see is that in beautiful oscillation, these little dots are individual neurons, the 2 suprachiasmatic nuclei oscillate. That is to say, the PER1 gene expression oscillates in these 2 little structures. And this actually lasts for weeks to months. So even in vitro, this system is highly robust because the neurons are coupled. Well, once the clock genes have been identified in mammals, this was relatively late, in the late 90s, we and others were, of course, curious to see whether these clock genes are only expressed in the suprachiasmatic nucleus, which is the master clock. At that time, it has been thought that the master clock is the only structure where gene expression oscillates. So we have repeated experiments of others and shown that period 1 messenger RNA, as shown by this technique called in situ hybridization, oscillates in these little 2 dots, which are the suprachiasmatic nuclei. Beautifully oscillates. Well, what about peripheral tissues like the liver, the skin, kidney, and so on? So we took a time series of liver and using another technique, I won't go into the details, to measure these clock messenger RNA, we could show that not only is this gene expressed in the liver, and all other clock genes for that matter, but it actually oscillates in the liver. Now the question then arises of whether there are clocks everywhere. Whether there are clocks everywhere. This experiment doesn't show that because it's still possible that the suprachiasmatic nucleus emits signals through the blood, which makes these clock genes go up and down in the periphery without having actually their own clock. So in order to show there's a clock, one has to isolate tissues or even cells from the body and examine whether they stay rhythmic. This is exactly what a postdoctoral collaborator did in the laboratory. He took actually fibroblasts, skin fibroblasts that were taken 40 years earlier from a rat and have been cultured in tissue culture. And he measured the accumulation of a few clock gene products. And as you see here, they beautifully oscillate. But in order to see this oscillation, he had to be able to synchronize them. And that's exactly what he did. He added a high concentration of serum, which contains a lot of signals. And these synchronized these clocks, so he could see these beautiful oscillations. To show that actually each cell has its own clock, another postdoc, Emi Nagoshi in the laboratory, did an experiment in which he engineered a cell line that is kept in culture that expresses a fluorescent, yellow fluorescent protein, again under the control of a circadian gene. In this case, Referb alpha. She made the protein short-lived so it could go down again, and she made it nuclear because it's easier to see the signal when it's concentrated in the round nuclei. And this shows you what she got as a result. It's quite nice. You see that every nucleus is expressing these clock genes in circadian fashion, even in these dividing cells here. Of course, the cells move a little bit. That's why they twiggle around here. But again, you can follow these oscillations. They never stop. You can follow them until the cell is dead. So these oscillators in the cells are just as robust as the ones in the neurons of the suprachiasmatic nucleus. This experiment shows you that feeding cycles are very important in synchronizing the clocks. The peripheral clocks, I should say. So Francesca Damiola and Nguyen Minh Le, who's an Italian graduate student and a Vietnamese postdoc, did a really simple experiment. They just fed the mice either during the night, which is normal because they're nocturnal, or during the day. If one looks at the gene expression, in this case of PER2 messenger RNA, in the suprachiasmatic nucleus, these are these little two dots again, you see that the suprachiasmatic nucleus is not impressed by feeding cycles. It just keeps its pace. It doesn't care about feeding. However, if one looks at the liver, and for that matter, at most other organs, if not all of them, after about 2 weeks of daytime feeding, and they're fed during the day, they switch completely the phase by 12 hours. So now they're antiphasic to the rhythms they had before when they were fed at night. So the food is actually even dominant over the suprachiasmatic nucleus in synchronizing these peripheral clocks. So far, I have always pretended that the SCN is synchronizing peripheral clocks. But this has never been formally shown, actually. And the problem is the technology was lacking. Because if you remove the SCN, if you destroy it, then the animal has no phase anymore. It runs in little bouts, but doesn't have a 24-hour rhythm anymore. So you can actually not synchronize the peripheral organs. And it's very difficult to do a time series if you don't know the phase of the animal. You don't know whether it's active if there is a rhythm in peripheral organs, whether it's active mostly at 2 o'clock or at 7 o'clock or whatever. So for these, we absolutely need to show whether it's actually required for synchronizing the clocks. We absolutely need a new method that allows us to record gene expression in endogenous organs in real time during weeks. We have spent a lot of effort in doing this, And I would like to give credit to Camille Saino, graduate student, Pascal Goss, a technician, who did these experiments in our laboratory. The machine was built by our extremely talented mechanical engineers, André Eliani and Yves-Alain Boget. And we got the help from two photonics physicists, Luigi Bonaccina and Jean-Pierre Wolf from the Department of Applied Physics of our university. This shows you the apparatus they were engineering. Sorry, yes. So it is actually a light-proof cage that guides photons to the surface of a photomultiplier tube, a very sensitive device to count photons. It's about 1,000,000-fold more sensitive than our eyes. This is equipped with a feeding machine, so we can feed them throughout the day, or only during the night, or only during the day. And this shows you the water, which contains luciferin. So if you place a luminescing mouse into this cage, we can actually follow the emission of photons, and therefore the circadian rhythm, if there is any, of the peripheral organs. We got some very talented surgeons in Lausanne that are able to remove a tiny little piece of the brain that contains the suprachiasmatic nucleus, so they can destroy it. And if you measure locomotor activity, as you see here, it's chaos, it's not organized. In contrast to this mouse that has an SCN, which is beautifully rhythmic. But now we can also not only measure the behavior, we can measure the circadian gene expression in the entire body, or in particular organs, which I won't show here for time reasons. So this shows that if we do that experiment, and we do it with a mouse that we got from Joe Takahashi at Southwestern University, then we can see that each day shows this very beautiful, robust, high amplitude peak of Purge gene expression. It's a clock gene. However, if we feed the mice throughout the day, it's almost chaos. There's still a tiny rhythm left. We know why, but I won't have time to explain you. If we, however, impose 12-hour feeding fasting cycles, you see that even in the suprachiasmatic nucleus lesioned mice, we get the beautiful rhythms back, because now the liver clocks are synchronized, the body clocks are synchronized again. If we switch the rhythm, it's difficult to see here because too many peaks, but believe me that within 1 or 2 days, the rhythm changes. It actually goes much faster than in normal mice, because now the suprachiasmatic nucleus is not counteracting the effect of the feeding. So you see that this is almost chaos. It's actually worse than a symphony orchestra without director, because the directors, which in this case is the suprachiasmatic nucleus, which lacks here, but the musicians, they listen to each other, so they could still play in harmony. It's actually more like each musician playing a different room, so they know of each other and there is no rhythm. So clearly the suprachiasmatic nucleus is absolutely required to synchronize peripheral clocks. I will end by a few remarks about the system during our lifetime. As you can imagine, unfortunately, the circadian timing system is not different from the rest of our physiology. It deteriorates with age. So the oscillations, as measured in amplitudes here, how much they go up and down, they dampen considerably with age. And this concerns sleep-wake cycles, metabolism, and body temperature, and actually, for that matter, virtually all circadian functions. Another interesting aspect is so-called chronotypes. We have different activities during our spare time. Of course, if you have to go to work, we all have to leave early in the morning. But if you are on vacation or during weekends, we have different what's called chronotypes, activity periods. This can be measured by a parameter called mid-sleep time. So when you go to bed, like in this case here at 11 p.m. and you wake up at 7 a.m., the middle of that would be would be 2 o'clock. So you would be at 2 o'clock somewhere here. So now we measure these based on tens of thousands of individuals. Theo Ronneberg found out on how these chronotypes are distributed over the population, and he found there's a hugely wide distribution. So the morning larks, they have mid-sleep time even earlier than 2 a.m., whereas the night owls, the other extreme, even later than 9 a.m. So the chronotype changes during age, and this is particularly bad. Sorry, I shouldn't say this. Changes in the population, and this is particularly bad if one looks at different age classes. You see that 10-year-old children, they have mid-time sleep about 3.5. Of course, they also sleep longer, not just 8 hours. And then one looks at young adolescents and young adults, and this is largely different. You see it goes to about 5 a.m., and then with age it goes down again. I'm an exception because I'm still a night owl in contrast to most of my peers. So this is bad because particularly for adolescents and young adults, because they usually have to go to school or to work early in the morning when they're not very efficient. So for actually in high school or at university, it would be better to start the lectures at 10 a.m. rather than at 8. And I come to the summary. I've shown you that the mammalian timing system has a hierarchical structure with a master pacemaker, so-called suprachiasmatic nucleus in the brain, that synchronizes peripheral clocks in virtually all cells. This system participates in controlling the timing of nearly all body functions. The molecular oscillators function in a self-sustained and cell-autonomous manner. That is to say, each cell has its own oscillator that never stops working. The SCN synchronizes the peripheral oscillators via indirect Zeitgebers, rest activity rhythms, that, of course, drive feeding fasting cycles because you don't eat when you're asleep. These are the dominant Zeitgebers, but they work also together with hormones, for example, cortisol and body temperature, that are more directly regulated by the suprachiasmatic nucleus. The suprachiasmatic nucleus itself is synchronized to geophysical time, mostly through light-dark cycles, but the rest of the body clocks, they actually they're synchronized mostly by feeding fasting cycles. I've shown you at the end that human chronotypes vary greatly between individuals and change during aging. And I have indicated, without showing you the evidence, that the robustness of the circadian oscillations in activity and physiology unfortunately deteriorates during age. Thank you very much.
I had a lot of fun watching your presentation, also because it was the first science presentation I had watched 3 years after finishing my degree. And so I really enjoyed getting back into looking at the molecules and thinking about the experiments. So
Thank you.
I wanted to ask you, what are some priorities for you in experimental design? Do you like to start with the methods, or do you like to start by asking questions and then determining what you can answer?
Yes, that's a very interesting question, particularly for life sciences. Of course, we have to start with a question, and then we designed the methodology, and if not available, we try to make our own methodology. Like, for example, you saw this cage with the mouse where we can record circadian gene expression in real time in the living mouse for weeks to months. And we can not only do this for the entire mouse, as I showed in my presentation because of time reasons, but we can also show it for only the liver in the mouse. So we can look at it for, in particular, organs. So you ask a question, and frequently when you ask a question, then you get a different answer. So
That's
I believe
right.
that life sciences are mostly driven by, are mostly actually driven by experiments and the answers you get for experiments, rather than by a very defined question. Of course, you have to have a question before you start, but frequently you don't get the expected answer, and maybe the answer you get is even more interesting than the question you were asking.
Yeah, that's right. I can really relate to that. So much of playing piano is dealing with the unexpected, going into hall where you don't already play the piano, or what happens when you play a wrong note or you do something different than what you were expecting. So
Yes.
what I took away from your presentation was that the knowledge about the central clock in the SCN was known long before the peripheral clocks were known.
Yes.
Um, So presumably there were probably things that could be accomplished just using the central clock, like having a regular rhythm throughout the day. But what motivated the discovery of peripheral clocks, and why is it important or significant that they exist separate from the central clock?
Right. So in mammals, the only light sensitive cells are photoreceptor cells in the retina. So the light dark information has to be transmitted to the central clock. And it's quite well known how this works. It would take too long to explain it, but this is quite well known. And the central clock then could either regulate everything by just regulating hormones, that would be possible in principle. But as soon as these clock genes were discovered, we and many other people were interested whether these clock genes are also expressed in other tissues. And it turned out that they were expressed in all tissues. Then the next question was, as I presented in the lecture, is this due to real autonomous clocks, or is it just driven passively by the central clock? And that's why we did this experiment first with cells. And we managed to synchronize the oscillators in the cells, and then the unexpected result was that they have just perfect clock, just like the suprachiasmatic nucleus. So the second question is, what is the advantage of that? And one advantage is that, for example, if the mice have, or we, or any other mammal has access to food at another time, the SCN is still driven by the light dark cycle, but the liver can now adapt to the new feeding regime. You know, in nature, it's possible that all of a sudden food gets available at another time. So the liver can actually adapt to that independently of the central clock. This would not be possible if the central clock just drives everything passively.
Yeah, I see that. Are there any organisms that have only one or the other kind of clock?
Ah, very good question. Of course, the circadian clocks even exist in bacteria, in some bacteria, or in monocellular organisms. So then you have one clock. For example, cyanobacteria. And then there are also metazoans, in other words, animals with many cells, and even vertebrates. For example, the zebrafish is a small semi-transparent animal, and all of its organs are light sensitive, even cell culture. So So the peripheral clocks can actually adapt to the light-dark cycle, even within the central clock, which is probably the pineal gland, not the SCN in this small fish. So yes, there are organisms that only have, at least where it's important to have peripheral clocks that are directly regulated to the light-dark cycle, and there are other organisms which, particularly unicellular organisms, of course, which only have one clock, so you can't say it's central or peripheral.
Right. One image that I really remembered from your presentation was when you had that mouse and it lit up in your little chamber. And it just reminded me of this video game that I really liked to play growing up, which is Pokemon. And the central character of that is Pikachu, which is an electric mouse, but his main characteristic is that he lights up and he's bright yellow. So I just really enjoyed that. So thank you so much.
Oh, it was a pleasure. So my question to Hilda is related to the H art event, in particular with H. You know, it seems that you have to have such a fast reaction with your fingers that playing piano would actually be more difficult with H. So my question is, if one takes a very old pianist like Arthur Rubinstein, who gave his last concert, I believe, at the age of 87. Did you feel that this concert was less good than the ones he gave in his 50s or 40s?
I think that's a good question. I mean, that really strikes to the heart of what constitutes the activity of playing piano. And that's actually something that's really up for debate. You know, I think when you're young, you can play fast, you can play loud. These things become your primary toolbox. But once you develop your technique more, you start to integrate a lot more different pieces so that you simplify the manner of your expression. And in that way, it makes it technically less demanding. So I bet actually that when Rubinstein got older, he probably felt that it was easier to play in some ways, even though his physical stamina might have decreased.
All right. So the second question is, what always amazed me is how the brain can actually process the information. You know, when you're a musician, particularly a pianist, is reading the music. The music gives commands for 10 fingers, I guess the pedal, the speed, and so on. And then on top of that, you have to give it a certain style that makes you different from other pianists. So to me, it is incredible how this works. And the question is there any brain imaging of pianists while they're playing the piano and reading the music, either reading the music or actually playing it by heart? Both would be interesting.
I actually don't know if there's experiments done with people playing while reading music, but there have been experiments contrasting how people play when they're playing something by heart versus when they're improvising. And the studies do show that different parts of the brain light up in fMRI experiments. You know, they have you go in the tube and you have a little keyboard out front.
All right.
But in particular, when people are improvising, some scientists have found that the regions involved in inhibition really turn off, which implies to us that there's some kind of a flow state going on when we're improvising. And hopefully, you know, when we play something by heart, we can also improvise with the parts of the music that aren't just the notes.
So this raises actually another question, because I thought that improvising is something particularly to jazz. But you tell me that you don't mind listening to jazz, but you don't play it yourself. So to what extent can you improvise in classical music?
I think that's a great question, and that's really relevant right now, because right now it is a little bit after the peak of historical performance, and one of the main tenets of this practice was the practice of historical improvisation, like improvising in the Baroque style or in the classical style. This was how they composed and how people became masters of their instruments. For us, we can improvise with ornaments and with dynamics. But for them, they did the whole thing. They did the fugue, they did the prelude, they did the whole suite. So it's something that as musicians, as an industry, like as a field of study, we're really, that's a topic we're all working on right now.
All right. And a very burning question for a biologist and geneticist like me is that to what extent is the talent for music, for example, playing piano, is that genetic? In other words, is there a hereditary basis for playing well or not play, or for learning to play and so on? So the question is, are there identical twin studies where children with the same genome have been separated, and to see whether both of them develop an interest or a talent for music? And the second part is, that's much more easy to do, I think. Are there cases where very famous musicians came from families where they never were exposed to music?
Yeah, that question about twin studies, I actually don't know any literature on it, but I do have some experience with just my friends from growing up. You know, there were twins. And oftentimes one of them just had more of a willingness to participate in it. And that seemed to be the thing that set them apart. You know, both of them were really talented players. These were two violinists that I knew growing up. They were both very skilled, very talented, almost like prodigies. But it seemed like there were a lot of other factors, social factors, family factors that really encouraged one to go in one direction and one to go in another. There are certainly lots of musicians, including myself, who don't have families who are musicians, or musicians in the rest of the family. But it's very unlikely that people, if they're not exposed to it, that they should go into it. And usually that exposure first comes socially, whether it's through the family or through a family friend.
Yeah, this is funny because, of course, in science it's completely different. I myself was never exposed to science because my father was a sculptor. He was an artist. So you don't need exposure to do science.
Yeah, I mean, I think the issue of early exposure is something that a lot of American classical music institutions are really interested in working on right now. They have a big interest in exposing lots of kids to classical music by bringing artists to schools, to after school programs, and various community events, just so that people can see what's out there. And that seems to be an important step for a lot of young students.
Thank you.